morning, everybody. It's time to hear about some neuroscience. So I'm Amy Robinson. I'm the director of EyeWire, and I'm just going to do a few words before I throw it over to Will. But before we kind of dive in, I want to just kind of take it up to kind of a more high level to really appreciate just how complicated the brain is. You know, if you look at the world behind us and the world in front of us, they're small compared to the world within us. That may seem weird considering the vast amount of data and how much larger the Earth is, than the little organ that's sitting inside your skull. But from a neuroscientific and from a philosophical standpoint, the brain is nothing short of immense. It's arguably one of the most extraordinary things in the known universe. And when you really think about it, it's this 3-pound organ made of 80 billion neurons and some other types of cells that makes you you. It makes you able to think and feel and learn and love and feel pain and persevere. How does the brain do that? It's one of the greatest mysteries of all time. And fortunately for us, we live in an age of exponential innovation in neuroscience. It's kind of a neuroscience renaissance, if you will. And it's affording us an unprecedented view into the organ that makes us ourselves. We're seeing advances in technology and imaging that let us look deeper and at higher time resolutions. But it kind of comes with a catch-22. Most of the new technology that we're seeing in neuroscience is being developed by neuroscience and biology labs, which are obviously full of neuroscientists and biologists, not computer scientists. So EyeWire came from a computational neuroscience lab at MIT, where we use machine learning to try to recognize 3D objects, neurons, from stacks of images. And we're taking an interesting approach. While the lab continues to improve the AI pipeline, we're crowdsourcing data analysis with an online game called EyeWire, where we have around 200,000 gamers from 160 different countries who solve these 3D puzzles, and as they solve the puzzles, they're actually mapping the brain. So that's all for me. I'm going to throw it to Will, who is our technical lead on EyeWire, and he'll tell you a little bit about how deep learning is changing the way that we understand the brain.
Hello, guys. Oh, cool. Didn't know whether it was on or not yet. Hi, so I want to talk to you about the pipeline we use to actually get our neuroscience data into our game, and a little bit how we do our machine segmentations. And so that's why I call this talk Pipeline to Neuropia. So Neuropia is that place far distant in space and time where we actually understand the brain, and whether it'll be a utopia or a dystopia will be up to you guys. But I'm going to show you a little bit about how we're going to get there. So my name is William Silversmith. I'm technical lead at EyeWire. I work on the game, and I have a pretty general understanding of the whole pipeline that brings us the data, how it gets reconstructed. I'm not actually a machine learning expert, but I have enough of an understanding of the process that I think it'll be very interesting for you guys. So first I want to talk about the scientific problem that we have. So it's very difficult to see how neurons actually connect to each other. All these processes happen at a very small level. We're talking tens of nanometers, sometimes even a little smaller for some of them. There are other strategies that people... So the thing that we want to do is we want to see how neurons connect to each other. We want to see how microcircuits work within the brain. The specific organism that we're using is a mouse. We're investigating the mouse retina and how direction selectivity occurs within the retina. The other thing that we want to do is we want to create a classification system based on anatomy of the different kinds of cells in the retina. Previously, and there's a long and storied history of all these different techniques. People use genetics, they use physiology, how neurons spike, and they also use the morphology, the structure of the neuron, in order to do that. But there's at least one paper where our collaborators were able to find a new type of bipolar cell, which is one of the kinds of cells in the retina, just based on the anatomy, not based on these other indicators. So there is promise in this area. And being able to describe new cell types allows you to formulate new scientific questions for you to ask. So I just want to give you sort of an idea of what this sort of looks like. So this is a fellow that used to be at our lab. Here's a silicon plate. We've attached lots and lots of little bits of mouse So I'm not totally sure. This isn't the same data set that we were using, so I'm not sure what this data is, but it's neuro data, and it's very similar. And so you can see we get very, very small. So our data set, which was provided by some of our collaborators in Germany, is referred to as E2198. It's a piece of mouse retina. It was staged very atypically. So most stains of cells, they stain the internal organelles. So you can see things like mitochondria and nuclei. You can't see that here. We used the stain, or I should say they used the stain that only fills the extracellular space. And so very often, so you can't see anything on the inside. And we did this, or they did this, and we used it because this was cool, because it's easier to do machine learning on this, because you don't have all these artifacts and things inside the cells. However, that doesn't mean the data are perfect, because you can see, say, right over there, that little dark blob inside the cell is a cell that got punctured, and the ink started leaking into the cell. So nobody's perfect, right? So to give you a little bit about the dimensions of this data set, it's about 1/3 of a millimeter large. There's another slide coming up that'll give you more context. But each pixel in the data set along the X and Y dimensions is 16.5 nanometers, and there were cuts made in the Z direction using a diamond knife, and that was along 20. Each of those cuts was 23 nanometers. The guys in Heidelberg, Germany, they're cool guys. They like to make their own equipment, and so they have this setup because there are other setups where they have like people are doing slicing and it's going on a conveyor belt and they're like scanning these things, but there's all these like twists and turns and you have to do all these complicated image alignments, and it's particularly hard to do that in the Z direction down the stack because it's shifting. So what they did is they had a setup where they fixed their sample. They encased the retina in resin, and then they slice it, scan it, slice it, scan it. So that way they know it's not moving around. And they referred to that as serial block phase electron microscopy. So here's an overview of our data set. You can see that here's the very top layer. So this is 1/3 of a millimeter. Here's the very top layer. You can see landmarks within the data set. Here are veins, cells, and here's the area that we're actually interested in investigating. So this is not all the layers of the retina. This is only what's referred to as the interplexiform layer and some of its surrounding layers. Plexiform meaning spaghetti. So you can see all these cell bodies. Some of them are circled, and there's the spaghetti. So why? Why the retina? It's kind of a weird thing to look at, right? What everyone's interested in is, say, like cortical networks. Like, where does thought come from? Well, I sort of think that the way that you get there is you sort of like, when you're doing, say, a mathematical proof, you start from axioms, you work your way back, and eventually you get to the crazy abstract stuff. So the retina is cool because it's more complicated than, say, like than lots of other parts that are easily accessible. It's very cool because you could just shine a light on it and it turns on. It's super digital, right? And the other cool thing is that if you investigate a very small part of the retina, it's mosaic. Some of the neurons like actually tile it, and others have like these repeating circuit elements. Because that's why when I wave my hand around like that to you guys, it doesn't look like my hand is getting distorted based on the angle because there's all these repeating circuit elements, which is kind of similar to some CNN implementations. So here's what it looks like when you put everything together. Here's what our data set will look like, and here's what we want to get out of it. So here's just a small chunk of it, of volume, and here's the slicings. We're trying to follow a particular neuron down through the data set. And there's 2 neurons, and we want to see how they touch, how they synapse onto each other, etc. as you can see, this particular video does not have that atypical state I was talking about. You can see vesicles, which contain the neurotransmitters that will be transmitted to its postsynaptic cell. So what does the pipeline to neuropia look like? First, acquire images. 2, question mark, 3, profit, I'm kidding. Second, you align the images. You apply convolutional neural network to segment the images. Actually, it produces a 3D affinity graph. And then you run watershed to turn that affinity graph. So watershed's another algorithm. I'll discuss it a little bit later. And you turn that into final segmentations. Omnification is an internal process we use to get everything to work with our internal servers and things like that. And then finally, humans go and take those segmentations and validate them and turn them into like full-blown neuron models that we can actually use for science. So I was discussing how image alignment is pretty tricky. So in 2D, it's actually not so bad. In 2D, you could go and so in 2D, you actually have overlap from the electron microscope scans. And so you could actually find features that directly correspond to other features on other images. And so it's much easier to do that. But when you're doing that over tens of thousands of images that are all adjacent to each other, sometimes you still have to do some manual correction. And it's much trickier in the z direction because the image is shifting. But we're able to deal with it, but it does require some manual labor in addition to algorithms. Next slide. So I want to talk a little bit about getting into our actual convolutional neural networks. So first, I want to talk about our objective function, which was malice. So this was a paper that was published in 2009, where so before people were using pixel boundary error in order to do image segmentation. So I'm not totally sure if maybe this is like something new or not for you guys. But what malice does is it looks at the rand error for when we're segmenting all the neurons. So with the pixel boundary error, if you could cause one large area to leak into the other, even though the pixel error would be small, with rand error, it checks to see what proportion of pixels are grouped correctly next to each other. And there's some other tricks to it where it'll look within the patch of the CNN, and then it'll find what's called the maxi-min disaffinity path. And so it's the maximum minimum disaffinity path, and then you can tweak a threshold to create partitions within the graph. And you can sort of see that sort of what's going on over here. So this slide over here, affinity graph 1. So these different sections here would represent different neurons. This would be like something the CNN generates. And then this would be the ideal segmentation. This is real bad because now we've destroyed our connectivity graph because we have a merger here. But we're allowed to lose internal edges within our affinity graph. Yeah, and it's one interesting thing to mention about it is that we use connected components to define the segmentation because we want the important learning to occur within the thing, within the machine learning algorithm. And I think I have to speed up a little bit because I'm running out of time. But so ZNN is our custom implementation of a convolutional neural network. It is it's multi-CPU, not GPU. So the author of it told me that it is competitive at around 18 CPUs to a modern GPU. It has 5 hidden layers, uses malice. We use 7x7x7 receptive fields, cubic voxels. And yeah, it was trained to convergence over months of CPU time, and it was trained in 2012, 2013, and we're training it on a new thing. Watershed is the way that we actually unite all the pixels together to create the segmentations from the affinity graph. running out of time, so I will skip over this a little bit. But suffice to say that it goes from here. Here's what it does by default. You do some post-processing, and you get something that looks like that. And here's the final segmentation, or rather, here's the ground truth segmentation that it's supposed to match. And so the problem is it's not quite good enough and out of time. So I'm just going to finish with a video. And here's our AI accuracy versus our player consensus. Sorry about that. And that's just sort of a synaptic junction that we found with our final models. All right. Thank you very much, everyone. Let's move this here.